latest version. Okay, so this is what we are going to do related to this mindful alignment, mindful learning. So we are going to download this example MATLAB code and go through and to look at this Laplacian eigenmap application for this Swiss roll example data. So you can look at the source code to match the lecture and algorithm description and see which one is which. And then we will download this dimensionality reduction toolbox. And this toolbox implements a lot of these manifold learning algorithms. So we will play with this and using the GUI graphic user interface the toolbox provide to explore more example data rather than just a Swiss roll. Swiss roll is very famous example data to show this nonlinear manifold structure. Then we will visit example geo-accession record, and here we are going to use this example to show you if you really want to download a published data set, but the authors or the data generator, they didn't provide sample level matrix files, so we cannot just directly from the toolbox to import the each sample's matrix. But normally I just give up because that means authors didn't know how to upload the file correctly. But if you really, really want to use their data, and if they provide this irregular format of data, I will show you how we can download and using a little bit of scripting, coding, okay, to import the data from the RDS file. So in this example, the data was put into the R data file, so we can see how we can import that into MATLAB, so you can process downstream and analyze using the way that you're familiar with and compare the published results. And then we will look at this magic. We mentioned the magic, and that's for imputations. We do have this implemented. Should be ML. So there's a typo here. So I believe this should be right. Okay, so there's a function in run.ml, the magic. So we will look at this and see what's the impact, what's the effects after you apply imputation to a sparse matrix. In this case, our sparse matrix is gene expression, a single cell gene expression matrix. Then we will look at tensor data. So we try to do this SVD for 3D, okay, 3D array. So you can define 25 by 25 by 9, something like this. So this is a tensor. Then we will download a tensor toolbox and see how we can use this to decompose a 3D array. And if we still have time, I will see, okay, there's a function to do cell subsampling. And if you check the publication, you will find that this is called a geo-sketching algorithms to help you to subsample cells from, say, you have 1 million cells you cannot handle on your laptop, but you want to subsample 10,000 cells out of the 1 million. And how can you do it more effectively to select cells that's most representative to all the cell type? If you do purely random, so you may sample a lot. If you have a fibroblast, say in 1 million you have 900,000 fibroblasts, and only very few are other cell types. But this geo-sketching will help you to not only oversample the fibroblasts, but also considering other diverse cell types, even though the original cell number in the real cell type is small. This geo-sketching try to spread their attention to those lower frequency subtypes of cells as well. Okay, so let's download this M file attached here. I hope you can right-click and download this M file. This should be in your download person under your personal folder, then in the download. If you right-click these 3 dots and download, and then click that folder, you should have that in here. So this is my download folder. It's my in C drive user, my username, then downloads. So that's a place where this download file is located. So we want to do a CD. So CD is change directory to that folder. So you put that in there. Okay. Now your working folder is the one you CD to. Okay. Now let's find that one download. So this is our source code. This file. So we will try to quickly go through each section. So this, for example, will create this spring row data. And randomly generate them. This is the 3D data, X, Y, Z. Okay, let me know if you have trouble to locate this file and open this, because I want you to see the source code so we can run each section one by one. In your Mac machine may different. I don't know how to operate it. Same idea. Okay, any? So we are going to highlight the first section. So this is first section. This divided by this double percentage sign here. So this is one section from line 1 to 11. If you run this, the whole thing will just go through. But we'd better to run section. So you click this button here, step by step. So my cursor is right now in this section. So if I run this section, I will have all the source code executed, and then I get X, Y, Z, and the data. So there's 3D, right? X, Y, Z. 2048 data points. Then run this section, you should have a figure, a 3D figure. Okay. So those X, Y, Z was not generated by random. It's follow some kind of times. So it starts from some here and then go through. So that's why using its order, and you can color them. Okay. So because here there's no information about which X1, Y1, Z1 in front, but only by the order. So the three columns, first row, second row, they are ordered. So that's why you can create this a series of this color map. Okay, the rainbow color map. Now let's do, so here there's K and run this section, nothing should happen. Let's set up this. This part is compute, so-called P distance. So you have 2048 data points, then this will be every point to all the others, the pairwise distance computation. So if you do this, you should have dt. So dt is here. Distance already it's changed. Top ones. Yeah, I think that's all. The immediately you generate from a 2D, from 3D, and you embed this into 2D. The different so this looks like it's a linear. So we need to read out each step what they are doing. One, that's original. Graph Laplacian, I believe here is doing some normalization of the. So it's weighted. And this is a kernel, so you change, and by adding this weight, and get this eigenvector. So anyway, so this is a. I don't believe this is correct code. So it's supposed to have the. So this is the last one, and they put the results like that. So that's before the transformation, before the dimensionality reduction, and that's the results of the Laplacian eigenmap algorithm. So this is just show you the using created, constructed example to visualize. So I'm pretty sure this is the linear. I don't know where this come from. I have to read the source code more carefully. So there. Looks like it's using different components of this eigenvector. So looks like this is a 2, 4, and this is using 2, 3, and this is a 4, 3, and they decide to use 2, 4, and this 2 to do this embedding. Okay. Okay, so let's move. Now we need to visit this page. So you're going to do the download again. Then remember where you download the toolbox, we may need to unzip it. So this is a toolbox developed by Martin. Okay, he's the author for the Tisney, if I didn't remember it wrong. So OU map. So one of this algorithm he developed. So in the same time, so he developed this comprehensive dimensionality reduction toolbox. That's of course including PCA, and also many for nonlinear purpose, as you see this long list. And this should be in MATLAB, so we can download. Click here to download the toolbox. Okay, so what you can see here is a toolbox which zipped the tarball, and so you see the tar.gz. So you need to come to this page, to this folder, the download folder, and right click to extract using this 7-zip. That's what I can get. So extract here. I don't know how you do it in on Mac machine, but Mac machine should have a better support for this GZ file. But I extract this first. This will remove the GZ extension and give you the toolbox here. I may need to do another round. Let me see. So what's the compressed folder? Its trouble is I don't see the extension of the file. So maybe this is a tar file. So I do another round of extraction. Extract here. Yeah. So after 2 rounds of extraction, so I changed. Let's see. The toolbox is. So there's a toolbox name. Then you have a TAR. And then GZ, so this is the file name. So the first round, I do the extraction, I remove this and change this into toolbox TAR file. Okay, the tar file is still packed, so I do another round of the extraction. Then I have a toolbox folder, and all the files is under this folder. So 2 rounds of this. First round, I change this GZ file into tar file, and then after another round, I got this toolbox. So you need to fully extract or expand this until this become a real folder, okay? And then you can delete, if you want, delete the zipped file, or so only have this folder. So this folder, if you goes into this folder, you can see all the M files in here, okay? So now you need to know where you unzipped this toolbox. So copy this folder name. So I'm going to come back to here. Let's remove this, clean the folder. So I will do a CD, change this to. Okay, I changed my working folder, goes into this toolbox, so I can see the source code, everything here. And I delete the old variables in the workspace. So this toolbox is organized not only one root folder, but also have two subfolders. So we need to call the function within the set subfolders. So, but they are not accessible for MATLAB. So we need to right-click and add this to the parse. So add the parse to the select folder. And then you can see after the hard to see after. So you can also do this, add a folder, add a parse. Say, okay, I have a folder called geo. So I will add this. I will also add another one, this technique. Then I finish the setup for this. If you decide to use the toolbox from outside the toolbox folder, you have to add this folder as well. So you have to add this root folder as well. Okay. Anyway, you just have to let the system know. Okay, when you need a function, you need to search the function name under those folder after you add them in, then system, those functions become available for the system. You can see, okay, in the technique, that's where the algorithm is implemented. Technique. See this, try to find the PCA. Yes, PCA is here. So if you look at his PCA implementation, you have input, and you map the results, mapping. And this is compute the mean, and then centralize the data, basically. You have a mean, then the original minus the mean. And this is a place where the covariance matrix is computed. And then, according to the algorithm, we have to do the eigen decomposition for the covariance matrix to get the eigenvectors. Eigenvectors will be used, so sorted by the eigenvalues, will be used to rotate your input matrix, okay, your data. You use this as a rotation matrix, right, to get the rotated. So that's a implementation for PCA, but not the one we really need to play with. What we will do is to do GUI. So if you look at here, you have a DR GUI. I hope it can run in this new system. Okay, so this will bring up your small dialog window. So this place, you can load the data and select algorithm, and then to run. So I will load example data. If you start with a Swiss roll, and that's a place, will generate this, and you can look at the Swiss roll data generated. And then there's some function which not comparable with a new system. And then there are optional, maybe let's ignore this, and we'll directly go to the computing mapping. So that's where you'll select different algorithms, PCA, LDA, Laplacian, so isomap. So let's try isomap first. That's very simple. So isomap is to use so it's completed. Then you click this scatter plot to show the results. Okay. So that's how this small toy can be playing. Load the data and then found 3D cube. So this look at the data. Okay, so this pretty much like a single cell embedding. Then do a mapping using from this. So we'll see. Done. Try another one. So let's see 20. I don't know. Helix. Yeah, so something like this, and hope if you do PCA, run PCA, you get the results, which are not bad. Plot. So this is a results with different algorithms. Okay, so this is slightly old toolbox, so there are a lot of functions which not running properly, and so take efforts if you want to make the comparable with a new MATLAB version and the slightly change of those syntax, and sometimes it's difficult to maintain those. But this gave you ideas. If you want to have look the source code about a lot of this dimensionality reduction technique, then the toolbox provide you have this resource. All the algorithms have been implemented using native MATLAB language. And they're all here, okay? You see some of those compiled versions. So this place is to, if you look at the CCA collection data, those are some function which developed in C and then compiled into the MATLAB. So if you call this function, then the source code, the MATLAB will be calling a C program doing the job. So in theory, the C will be more computationally efficient than MATLAB itself. And it's a language which we believe is some low-level language, but you need time to code yourself. So MATLAB is a high-level language. You can do PCA, do a lot of things. But if you code in C, you have to take care of all the pointer, how the memory is handled, everything else, which more complicated. But some the procedures, we do need a quick algorithm that can implement in the low-level language to make it more efficient. Okay, let's clean up this environment, and we can switch back to any working folder, and then clear all. Okay, so we finished the first two items. Now I'm going to show you this for example, okay? You know, normally if we look at this geo database and the study, so this is study, you know, this is published in 2022, the single cell in the spinal cord injury mice. Okay, there are some neurons in there, and the paper has been published. And then there are 15 samples. So normally we should be able to do this, copy the sample accession number, come back to MATLAB, and then if you do toolbox and to load geo accession number, so that's what place you can put that number in. Okay, so this example is this. Okay, then if I put that in here and I get error message, so there's something wrong with this particular sample, and I have to come back here and check this sample 1 in the 15 samples, and then we look down, there's no file has been uploaded here. So the author didn't publish or correctly upload their sample level matrix. Yeah, so you shouldn't get anything from this sample if you try to load them from here. Yeah.
I think I can see the supplementary files though.
Yeah, so from this one here, right?
Yes.
Yeah. So that's what I will show you how to do this here. So instead of per sample, because they didn't provide per sample matrix, yeah, but they provide the processed in R. So we are going to download this. Just assume they have done a good job in R and then put their results by incorporating integrated data in 15 samples together in this. So we will take some time to download this file. Okay. Okay. So we can click. This is 1.8 gig. So it takes some time to download. Let's just click this and wait. So this file, they processed 15 samples together and upload their data file as a one study. Okay, data file. So so at least they provide this, so we can use this directly. So that's where this is like a special case. If you don't have a sample level matrix, we cannot load them, merge them here. Then we decide to download their process data. In this case, it's R processed. So as I said, this is like a special case, and sometimes you see the supplementary files that without this, but something else. You have to be knowledgeable enough then to know, okay, if they provide R process data, I will use, I show you, use this, import this R files. If they have Python version, the ScanPy will produce something like this. So this will be H5AD files. So depends on the situation. So in our case, we will download this, and we have to unzip it. That also is end with GZ. So take some time to download. Let's see what we can do in the meantime. Okay, in the meantime, we can move to use magic. Okay, how can we use magic? Okay, let's see. In this interface, we can start to load an example file. This example data is from the real sickle cell RNA-seq data, so you can use raw data. Okay, the process is okay. So what we are going to do is to export this data into the workspace. So if you do, we need X and G, the top three. Okay, so X is a sparse version, so let's convert it to full. So you can see them. So that's our matrix. And rows are the genes. So there's the gene names. The first gene is MRPL15. Second gene is LYPLA1. So that's the list of gene name. And this is the matrix. Okay, so now if I want to extract one of the genes expression across all the files, so if I do this, say 15, I found a gene. I random say 15, that's okay. Gene 15, okay, expression level. Let's extract the rows. So by 15. Okay, so this is the gene number 15 and its expression in 2075 cells. And we can pick another gene. Say 30. number 7 is okay. So let's do number 7. You can pick whatever you want. So I have two genes expression. You can check this is number 7, so it should be from the X matrix. See, I want to look at these two genes expression correlation. So if you do the scatter, number 7 here, 15 here. All right, so this is a correlation between these two. And so this point means there are cells, which gene expression level of number 7 is 3. And the expression level for this cell, gene number 15, is 13, okay? So you can take a look at G7 is this, G15 is this. Okay, so there's a scatter plot between two genes. What you cannot see is there are a lot of cells have 0, 0. Okay, so from this scatter plot, there should have a lot with 0, 2, 1, 1. So suppose you have a lot of cells which located in this considered as a box or bin. Right, so this scatter plot not very useful in this case because there's so many cells, they're sparse. So many cells have 0, 0, so many cells have 0, 1, 1, 0. It's highly condensed here. So you can do a correlation, COR. Looks like this two, no. So the correlation need to be vertical. So the correlation between these two is 0.2. Okay. So this is a Spearman correlation. If you do type, do the so by default it is Pearson, but if you do Spearman, you got a 0.17. So this tells us these two genes are positively correlated based on the raw data. Okay, but this picture is completely biased or influenced by the sparsity of the data because all the 0, 0, all the 0, 1s, they all because the sparsity is here. So make this calculation. I wouldn't say biased, but it's impact, okay? Because Spearman, based on the ranking and the Pearson definitely require. If you do Pearson, you get 2.1. They require the true value comparisons, and maybe with assumption of the normality. So you need to have the gene expression follow the Gaussian distribution by assumption. So what we can do is to do the imputated version. So we are going to do instead of using X itself, we're going to impute. So So see impute version. To run this ML and magic, and put your X in there. Okay, so this algorithm will compute another matrix, actually imputated version. So this version has the same dimension. So if you look at X and X impute, they are same size, but the value is different. Now we have X15. So it was from X itself, but now we extract this from impute version. So we extract the X GN15 expression from the imputed version. Then you do the same thing for the 7. Now, if you run the scatter plot between this imputed version, you get a picture like this. So this helps you to build a better correlation between two genes. Again, they are positive correlated. The computation of correlation coefficient will not rely on or influenced by the sparsity of those. Okay, if you do correlation between imputed version. You get a correlation 0.62. It was 21. Okay, 0. Spearman. So Spearman was much smaller than this imputed version. It was before the imputation 0.17, but now it's 0.63. Okay, so this gives you a better view of two genes expression correlation. So let's label the first. X label. So that will be G7. So just give name of gene here. And your Y label is 15. You can do a basic fitting by here by adding a linear. Okay, you can add some basic fitting, and here you can see the slope is positive. Yeah, I think we can take a break, and after the break, we should have, at least on my machine, this big file from the R process should be downloaded. We will see how can we import the data into the MATLAB. Okay. Okay, so now we know how to use the magic to do the imputation, right? Now come back to the file we download. This is our processed single cell data, and they already put everything into the same file, so there contains 15 different samples supposed to be. So we download this 1.8 gigabytes file, and also it's zipped. If you look at the extension of this is GZ, okay? We cannot read this GZ file directly. We need to unzip it to get this RDS file. RDS is our data science data file. So we will right click and then show more options and using 7-zip to extract here. So after this first round extraction, so this GZ file should be opened and saved into this RDS file. So let's see where's RDS file. So GZ file, you look different from RDS. RDS is this one here. So okay, although they have the same name. So the problem with this Windows system is I should have, let's see folder options. So if I do the folder options, I should, let's view and then uncheck this. So we need to uncheck this hide extensions for known file types. So if you uncheck this applied, you should see this file have extension with GZ, and then after you unzip this, you have this RDS. Okay. RDS is the file which are produced. In this case, is a surrogate package created. So remember where this file is located. We will come back to read this file. So now after you unzip the file, then we come back to this folder here and then clear our imputation example, and using this interface to click this to read RDS file, okay? So we will point to the file in the download folder and then find this unzipped version of this, okay? We'll click OK. So this will rely on your R system has been correctly set up, and you have a surrogate package installed. So this function will call R and read this file. Okay. Well, can I do this? Something happened here, so yes, same problem, yeah. Yeah, I think that's something changed in the interface R language. So we won't be able to do it today. So until next time, so fix this. So I believe there's one of the version changed. And yeah, so yeah, so I have to go back to fix this problem. So it's supposed to be able to read the R files into MATLAB automatically. So if you don't use MATLAB, you have to come back to the R environment and to see, okay, read RDS is one of the file, read R, the function, and then yeah, you have to find the file name and all this. So that's how you can handle it. So you have to have a full name and the file, and put this folder here, and then read this into the SE, for example. Yeah, so I have to do this correctly. So that's in R. SCE, you should have number of genes. So you have all this information in there. Okay. Yeah, it should have the name of all these counts. Yeah, so this is in R, so somehow you can handle that. The interface, after I make it correct, and should be able to get this all the information of the genes, or the count matrix, their annotation in there as well. So that's a place I have to go back and fix the problem. So that's in R, to read the file, and I believe there's some place here which cannot get X correctly. So yeah, so it's pretty much as a different version of the surrogate they use. So this is my interface between MATLAB to read the R data file. But the package, when they upload, that was older version of the surrogate. So supposed to use this different lines of the code to read. So I have to fix. So this is for the new version. New version. The new version have the layers of all this. So I make a change to read the new version, but the file we downloaded, older version of R surrogate created data. So this causes a problem. So I can, let's see. So if I make change here myself and reverse back to the older version, I should be able to demonstrate, at least for you. So you don't have to do it, but I will try to demonstrate after I change that. Go to the older version of the tools. It contains some metadata, so it's about annotation of the files. So we can use them later. Say, I don't want to do the annotation of files. It should have the this cluster name. So this column we can use, and that will be the sample ID and conditions. So those information we can use later, but that's the file I can load because I changed the source code back to the older version. And then if you read the papers they published here, you should be able to see the same figure. So this is their paper, so that's their data. So this one-to-one match, and also they are annotated based on cell type. This is not annotated, but we can load. See, load the data in. I'll quickly show you how to do it. So I'm going to save this to this. And then the T, so this is the metadata I got from their annotation. So let's see which is the cell type. So I will use this coarse clusters as a cell type. So now I just do this, say E, say cell type equal to T, because we have metadata from T and the call. So now I have the cell type. I can reload this into MATLAB. And then it's already annotated. So I import their data annotation to reconstruct the picture they had today here. So this will be very helpful for you to reanalyze people's published data. Okay, I just very quickly demonstrated it's possible, and if you know how to do it and don't know much effort, and just get their metadata in and this one cell here. But you can handle this using the way you're familiar with. There are too many genes, for example. Okay. Neurons. So if I do filter based on our standard QC, let's see how many genes and the cells will be removed. Yeah, so this is like a clean data. So I believe they generate, for example, the original, they have a 60,000 67,000 cells. Remember, there's like 15 samples put together. But based on my QC, there's only 27,000 high quality data. There are a lot of low quality data should be removed. So this picture, there's some problem. So as a very basic filter of the quality, because remember our cutoff really relaxed, even though I say stringent. I try to keep a lot of cells. But if their data cannot pass through that very basic filter and only left with this, so at least some of the cell type will not be very strongly supported. So microglia, they only have a few microglia if you consider there are 15 samples. So this is definitely not very good cell type. They can infer anything meaningful. They still have enough neurons maybe here. Okay, so this will give you a quick way to at least understand the quality of this study. Okay. Okay, let's clear this part. I will move back my working folder into here, for example. So now my working folder is the place where, like the root of the MATLAB working folder. So I'm going to continue with a tensor, okay? So MATLAB can define this high dimensional matrix, have no problem. So you know you can tap, say, okay, I have a X matrix contain random number and 3 and 4, okay? So you create a matrix 3 by 4, and then you can extract the position, say, okay, X 2, 2, this number here, right? We know this. There's no problem for you to construct high dimensional, okay? So like something like this. So now you create a tensor rather than a matrix. Now you can access the data in this tensor by like 2 to say 3. 3 is over the dimension here, so you have to be 1 or 2. So you can extract the data from, so that's the qubit, okay? So in this example is we create a Z. So this matrix, this tensor is 25 by 25 each layer, then you have 9 different layers. So when you click this, you only be able to see all of them. You only see the first layer, okay? Then you have different layers, up to 9 layers you define. All right, so you can define this. You can certainly increase this to another level, so fourth order and fifth. There's all possible. So it's getting bigger and bigger. I don't know if we can handle this scale. It's still okay. Yeah, so random number fill in this 0 high dimensional. So if you have a tensor data structure in MATLAB, for example, then SVD, for example, those will not working. And normally you have U as V, but because the function SVD only working to the so if you only see, I only need the fourth layer, it's okay. So now the Z become this should work because I extract the first layer of the second also work because they are matrix. So I can play this with one layer after another. But we do need a function to handle tensor structure. So that's where we need to install tensor toolbox. So let's click this to visit this tensor toolbox. Download its version 3.8. We can download a zipped file. Okay, so zipped file download in the download folder, and then to do the extraction O. Then it become a folder. So this folder inside, there's another folder. So that's where the package is. So you need to copy this folder name ID, the path. Remember, we need to add the path. The path I just copied is in here. So I download the toolbox and unzip it. Unzip it, then in the downloads folder, I create this unzipped folder and also this toolbox inside itself under the same name of that folder name again. But after I add this parse or the function inside this, tensor toolbox become available to me. So if you didn't add this parse in, so you only have this tensor object available, but now it's okay. So we need to read the documents to play with this, but maybe this tensor Z, I don't know if it works. Yeah, so some kind of this operations allow you to convert your 3D array into the tensor, this object which can be used by the toolbox functions. So remember, our Z is this randomly generated. Let's see how to use this function in the toolbox. So you can make this into a tensor TZ. So that's what I just did. To do this decomposition, the function is called CP, okay, from alternate least square function. So this is what calculate. Yeah, so we do need to convert. I believe we can convert this into the tensor Z. Let's try this, okay? The tensor Z here. No, yeah. So Z itself is a 3D array defined in MATLAB, native MATLAB. But we do need to have some transformation to make it become a tensor object. So I do have a typo here, so I need to change this. We need a TZ instead of Z to do this decomposition. So this function takes a tensor object and then to decompose using CP alternating least square method. And output is in the structure called M. So M is the composition, okay? 3D composition. So this 1, 2, 3. 3 small matrix. Their product, and with this lambda values as well. So let me draw this so you have a picture on your mind. So our input is 25 by 25 by 9. So this is our structure. $25 by 25 and 9. We have numbers. So we apply this. So this is called the TZ. So we apply this TZ into M using this function CP ALS. Is here, and we ask for 3D. So that's why they have 1, 2, 3 columns components. So this function to decompose this into 3 matrices. The first matrix is 25 by 3. And this will times with another 25 by 3. Another 25 by 3. And each of them have a factor 1, factor 2, factor 3. So the factors are this. You can consider them as the magnitude of the 3 components. So if we only ask for 2, and we can see you can change this into 2. Okay, you further reduce the dimension. So this 2, 2, 2 product. Or maybe not. So this is 25 times 25. So this will create 25 by 20. Here maybe it's a 25 by 9 by 3. maybe it's a 9. So let's check. So this is the first UV. Let's check the last one's dimension. So this is 25 by 3, 25 by 3. That's 9 by 3. Okay. So this is 25 by 3. At the end, this is 9 by 3. So if you use our matrix multiplication, so it doesn't matter 2 or 3. So you have 25 by 3. So that's our A3. And then 3 by 25 is our B. So the product between the A and the B, so transpose this, will be a 25 by 25. And here we have, I don't know how to do this. We have a 9 here. I don't know how to make this product. We have a 9 by 3 product. And this will reproduce 25 by 25. Okay. Anyway, so this 3 product will recreate this. So it's not like matrix multiplication, it's a tensor level. So if you do only need 2 components, top 2, you can get yeah. So the M will become this only 2 dimension, 2D, to approximate the whole product. So how can you, once you have those components, you have components, see, okay, I see ABC here, so that will be M mu1, U3. How can you recreate? Put them back. So that's the function, so just 4, okay? Put them back to reconstruct Z from M. Okay, now you reconstruct, you decompose them and reconstruct them back. Zm, then collapse maybe. The data, okay, the data is Z reconstructed. Data. Yeah, so we had a Z which is 25 by 25 by 9, and then we go through this decomposition and reconstruct. We come back with this, okay, Z constructed. This should be very similar to each other in theory. Okay. So why do this? Because just like SVD, we have a matrix and decompose into small matrices and only keep the most important components and reconstruct in order to denoise or learn the major components in our original matrix. Okay, but this is just extended to high D and not only the matrix, but only 3D array or even higher dimension you can do in the same way. So let's try, I want to try this, another layer, say another 2, another 5. Okay, I created this, I can do, make a tensor Tz, do the decomposition, M with 3 components. Remember, now we are handling a 4D array, okay? This should be able to do it, and then if you do Z4 and Z reconstructed, yeah, so the Z reconstructed also 25 by 5, so size Z, that's original size reconstructed. Let's go through this decomposition. So inside this M, okay, you have those components. And sometimes you want to visualize these components. For example, if I decide to use 1 and then do so that's my first dimension 3D components, and we may want to look at. I don't know if we can extract this. So plot. Yeah, so we can plot the components in the hold on. Second and the third. Yeah, so that just show you the relative values for this particular components inside this 4D data structure, and I look at the first dimension for that matrix. I decompose this into 3 components, and that's relative contribution for the components here. The input is purely random, so generated, but if you have some signals in your real data, you're probably able to see the change. For example, one component become monotonically decrease, another increase, so you can decompose those signals, okay? So hard to explain, but yeah, so that's the point. Okay. So I think we on our schedule, we have only one thing left. I'll show you how to do this subsampling, okay? We can use example data here to so this is a small data set, small enough, but see, okay, this is 2 million, for example, assume. Then there are subsample or 50% of cells. If you only want work with say 1,000 cells, because you have a very small machine, cannot handle 2,000 cells, then you just click this to see, I want to do the uniform subsampling. Then that will randomly select 50% of cells. And then if you have a particular, for example, this particular cell type, it has 200, then this subsampling will give you about 100 cells. But if you have 50, then you will have 25 only left. So this uniform subsampling will not take care of those. So you may end up with examples like this, and you don't have enough cells for this particular cell type, you can do anything for them. So let's load back this original process data. 2000 data you want to keep. So what's this cell type? Label the cell type. Yeah, so this is a macrophage. So in this sample, we don't have a lot macrophage. So if you do the subsampling uniformly, you will lose the subtype. So the idea for this 50% cell subsampling using this approach, geometric sketching. Let me see. The paper is this, and I hope it's working. Yeah, so I have to fix that again. So this is calling the Python function, and I don't know what the problem is. It's supposed to give you it will subsample relatively more for this real cell type. It's not working here. Yeah. Yeah, so but that's the idea, okay? So you can take a look at the paper, and I found that sometimes it will be useful, especially you have 25 samples put together, and then you want to only work on a quarter of those cells, and you do this subsampling twice, okay, 50%, 50%. Then very quickly you get some idea about the whole data structure, cell type, distributions. Then you want to work on the final results. You may find a more powerful machine to process the whole data set. Yeah, because of this problem here, I cannot show you exactly how this works, but yeah, that's the idea, okay? Okay, so this is for this week. Next week we are talking about the networks, okay? Networks also very important, so you can create the networks, for example, here, and by putting cells, and then there are different ways to compute those to get networks. Okay, so if you don't have any other questions, this is for this week, and again, next week will be the last week for the lecture, and before your